Hello, my name is Matthew. I'll be presenting on the topic of endotoxic mechanisms of lipopolysaccharides in gram-negative bacteria. And to start off, I just kind of wanted to provide a brief overview of the structure as well as the function of lipopolysaccharides, also known as LPS. The lipopolysaccharide is an essential negatively charged molecule found present on the outer membrane of gram-negative bacteria, and it's comprised of four distinct components. And here at the very bottom, we have lipid A. This is what's anchored to the outer membrane in the bacterium. It's responsible for the selective importation of water-soluble substances and also helps to promote a more stable intracellular environment within the bacterium. The hydrophobic component is found anchored at the base of the outer membrane and serves as the endotoxin, so this portion here. And right above lipid A, we have the inner core. This is a crucial integral piece for the overall structural integrity of LPS, as well as the membrane itself. And it's composed of a bunch of phosphorylated sugars that you can see here right above it. And right superior to the inner core, we have the outer core here in blue. And this facilitates the connection between the base of the lipopolysaccharide and the O antigen, which you can see right here. And it consists of just a chain of hexose sugars and kind of just connects the inner core lipid A to the O antigen. And right above the outer core, we have the O antigen. This is the terminal end-piece polysaccharide subunit of LPS. And this is what the antibody and response system will bind to. It's very antigenic and it will recognize it very quickly. And it's highly variable among other bacteria and serves as a kind of key source for identification of certain bacterial species. All right, and here we have an electron microscope image of an E. coli bacterium. It's still intact. And here you can kind of see this is where the plasma membrane would be, and right outside of it, you would have the lipopolysaccharides. You can't really see in too much detail just because they are so very small structurally, and it's very hard to fully visualize the structural component with electron microscopy just because of its incredibly small scale. All right, and next we have lipid A's endotoxic impact on the immune system. And here in figure 1, you can see that the endotoxin, which would be lipid A, is perceived by the TLR4 toll-like receptors on the host cell. These receptors are mainly found on sentinel cells or antigen-presenting cells such as macrophages and dendritic cells. So this is a transmembrane protein, which would be considered the PRR, or the pattern recognition receptor that would bind to the PAMP of lipid A, which is the pathogen-associated molecular protein. And TLR4 will in turn induce a signaling cascade response. It'll activate this transcription factor known as NFKB, and this will propagate the release of pro-inflammatory cytokines, as well as type 1 interferons that you can see here. And as a result, this innate response to this whole situation here, where lipid A would bind to TLR4 and result in this response, it may clear out the infection itself. However, the release of so many pro-inflammatory cytokines can be pretty concerning. It can result in a lot of inflammation and can be damaging to tissues with a lot of release of histamine. Granulocytes will release a lot of granules that can be very pro-inflammatory and actually be a lethal outcome for some patients. All right, and up next we have endotoxic shock, which is kind of the medical outcome of what you would see from a lipid A response and induction. So this is also widely known as septic shock in a lot of medical scenarios. Endotoxic shock can lead to a life-threatening decline in blood pressure, as well as manifests many symptoms such as fever, rapid heart rate, asthenia, chills, hypoxia, as well as internal bleeding in some cases. And it can be treated with oxygen therapy, antibiotics, in a lot of cases, IV therapy. And if it gets really progressive and severe, we can interfere with surgery. And lastly, how does this topic relate to the field of dentistry? So LPS, or lipopolysaccharide, can be the infective mediator for many bacterial species, such as Porphyromonas gingivalis and Prevotella intermedia that exists in our oral cavity. These bacterial species can cause diseases such as periodontitis, which is the inflammation of the gum tissue. So these mostly anaerobic bacteria will release LPS sometimes when they die, and it will result in large scale inflammation across the gum tissue. And it can result in a lot of discomfort and inflammation around the gingiva. And in some cases, it can infect the tooth and result in pulpitis, which is the inflammation of the pulp chamber itself. And this can be very painful and very uncomfortable for a lot of patients, resulting in a lot of acute tooth pain. And lastly, the lipopolysaccharide plays a primary role in the synthesis of biofilm. So LPS kind of gives it the adhesive properties to kind of initiate the proposition and production of lipopolysaccharides in the oral cavity. And lastly, here are my references. Thank you.